Welcome back to Air Quality Matters. We already have the tools and knowledge we need to make a difference to the quality of the air we breathe in our built environment. The conversations we have and how we share what we know is the key to our success. I'm Simon Jones, and coming up, a conversation with Janet Price, Chief Science Officer at Visium. If you caught one of the early One Take podcasts, I'll include a link in the show notes. We covered the promising technology of far UVC in managing airborne infection risk. Since then, I've been keen to follow up with a deeper dive into this fascinating technology, and one I don't know a lot about, to be honest. And so, myself and Janet Price from Visium started talking. Janet is an accomplished scientist directing R&D in the tech startup space with 11 years of research experience, 2 of which were spent overseeing cutting-edge UV research. She understands this space very well. She received her doctorate in molecular, cellular and developmental biology from the University of Michigan, where she specialised in microbiology. Spanning 3 separate collaborations, her dissertation research earned her recognition as a Rackham Pre-Doctoral Fellow. She is an accomplished science advocate with experience speaking to broad audiences, from invited lectures for specialists to hands-on presentation to inquisitive young minds. This is a fascinating 101 on far UV, what it is, how it works, its potential and limitations. Janet is a great communicator with deep knowledge of this technology. I think you will find the episode really fascinating. I certainly learnt loads. Now, Visium are mid-brand change from Lit Thinking, so if you're confused about who I'm talking about, that's the reason in the podcast. Please don't forget to check out the sponsors in the show notes and at airqualitymatters.net. This is a conversation with Janet Price. What do we actually mean by far UVC? And in terms of an air quality podcast, why does it matter?
Far UVC is a really special area of light. So if you think about our electromagnetic spectrum, we have our visible light and we have those wavelengths that come and fall outside of it. And some of it we're really familiar with. UVA and B are kind of near our visible spectrum. Those are what we get from the sun every day. UVC is a little further out. We get our 400 some odd is our beginning of our UV spectrum, but at the very far end of what is considered UV is UVC. So this is a short area of light. It's 280 to about 200 nanometers. And these are wavelengths that are known to be germicidal. So as we get closer to these high energy, this 222 is kind of the sweet spot for right now. It's able to interact with both DNA and RNA. So we're messing with what really constitutes the knowledge center of cells and viruses, all the way to protein. So now we're having a direct impact on the things that are alive and able to impact our health. So we've known that UVC in all of its wavelengths have had this impact for hundreds of years, but it's just now that we're getting the technology that allows us to harness it more efficiently and put it in places that we had never dreamed of it being able to do before.
That's interesting. So just to step back a tiny bit, when we talk about wavelengths of UV light, and we're talking about spectrums from UVA and B, and anybody that's got a teenage daughter is informed what the UV index is every day because they're
right.
just obsessed with nothing else, right? So on a spectrum, as we go down the numbers towards 220, so at the 222, which is what we're talking about with far UVC, does it become something else when you fall below 200? Is it moving more towards the visible wavelengths, or is it moving further away from the visible wavelengths when we're going up and down this spectrum, if you like?
The smaller the number, the further away we're going for it in this direction, right? So the electromagnetic spectrum has visible kind of right in the middle, because that's how we think of things. Our important stuff's in the middle. So as we go towards far UVC, these are getting to be very short wavelengths. And the reason that this is important is because long wavelengths penetrate more deeply into our tissues. That's why UVA and B are known to interact with our skin and can penetrate very deeply, whereas UVC and far UVC, we're getting less penetration, so we can now use it in places where people are present.
Okay, so like a lot of wavelengths, and I think people are familiar with this in the visible light spectrum. It's why fog lights are red, because they have a lower wavelength than white light or blue light, and therefore can penetrate through fog and particles more easily because they're flatter, I guess. So I'm guessing at this the much narrower spectrum, where you have much faster or peakier wavelengths, that they're impacted by stuff more, so that they have more potential, I guess, but they get blocked. Is that am I completely off the mark there?
No, you're pretty close. So the energy of these shorter wavelengths is closer to the energy in the bonds that are happening at the molecule level. So if you're thinking of the carbon-carbon bond that gives us life, if you're thinking about the phosphate backbone of our DNA, now these are bonds that this high energy can interact with. And that's exactly how it all works, is it gets absorbed by those bonds. And once that energy is gone, the energy of that wave is gone. That wave doesn't exist as it gets deeper and deeper into a tissue.
Really interesting. So is far UVC a naturally occurring light in our atmosphere? Is this something that we are exposed to naturally?
This is a really interesting question, because it does occur in nature. It just doesn't occur on Earth, right? So it is produced by our sun and other stars, and it travels to Earth. But this particular wavelength, like I've mentioned, because it gets absorbed by all the bonds that are life on Earth, it gets absorbed entirely by our atmosphere. So there isn't any UVC wavelengths from the sun hitting the surface of our planet. So all the UVC devices you see on Earth are the only source of UVC at the surface level.
Really interesting. So UVC A and B and the stuff from sunlight that does impact us, my teenagers are obsessed with, is able to get to the surface, but UVC is not something that we actually are exposed to in any capacity at all on Earth, unless we create it ourselves.
You've got it.
Okay, that's interesting. So what's actually happening? What when we say far UVC, let's start there. Actually, when you say UVC and far UVC, there's a difference there, isn't there? So we should be careful here. We've been talking about UVC for, as you say, hundreds of years for pathogen control. We go back to the Welleses and some of their early studies. Those that listened to the Carl Zimmerman podcast with me would have heard us talk about that early research. So we've known about UVC for a long time and its potential to have an impact on biological matter in the air. But far UVC specifically and UVC, is that a fairly recent invention, or was it far UVC that effectively the Welleses were using back in the day?
The delineation in language really happens because we're getting better at telling the difference in these wavelengths. So the light bulbs that the Welleses were using were mercury bulbs. They make broad spectrum UVC. So it's a bunch of different wavelengths within that 280 to 200 span. So you couldn't necessarily say exactly what wavelength is doing the most work, but together they're all having an effect. So now, as technology has gotten better and better and the way that we're producing these light wavelengths have changed, we're now able to get narrower peaks of light. So we can say this is something we can make with LEDs. So there are 260 LEDs that are very commonly used in water purification, right? Because they are kind of the sledgehammer of how to have an effect on something. They are very potent. They're really not supposed to be shined on people because they'll be just as potent on us. Where now we're starting to realize that there are different levels of what these lights are doing. So these higher wavelengths, the 265s, the 234s, they are more affecting the DNA and the nucleic acids. So think about the brain of the cell, telling how it's able to replicate itself. It's kind of scrambling that up. And we can get more into the specifics in a bit. But when we move to the far UVC, not only is it having that effect on DNA and RNA, it's also having an effect on protein. And because that has an entirely different pathway and the way that these lights are absorbed is a little different, we're starting to separate them out just so that we can really study and focus into how these lights are interacting with the surface or a pathogen differently. So it's probably only been in the last 30 or 40 years that we've really started to see far UVC, so these wavelengths that don't penetrate as deeply but have an effect on protein, really come to the forefront.
Okay, so it's that ability to be very specific and precise about what you're producing. Is that quite difficult to do? Is when you create light in this wavelength, the kind of things that produce it, which we'll talk about in a bit, can they have the potential to drift over time or deteriorate in their effectiveness, or can the wavelength change because of certain conditions? Is it quite hard to produce that specific wavelength of 222 and deliver it in the right way?
Yeah, so right now, the most pungent way, I want to say, of producing this light is the same way that we do neons, right? The idea that if you put a current through a gas, you will reliably get the exact same wavelength every single time because it's a characteristic of that gas. So before, like I said, the Wells used a mercury bulb. They had characteristic peaks, but it was a couple of them because mercury purity and whatever other gases are trapped in there, they'll all have their own wavelength. So what's been used to make far UVC 222 is krypton gas. So when you put a current through these krypton modules, you get a very repeatable peak at 222. Now, this will change once we get something in an LED, right? Where we can have even more narrow, very particular bands, but it all has to do with that technology. And it's under a lot of research. There are a lot of universities that are doing really great things in these semiconductor spaces, but the output's just not there where we can use it commercially yet. So right now we're going to do the old school way and put some energy through some known gases.
Interesting. And is there a power element to this as well, in the sense that you can produce that wavelength, but the amount of it that you can produce is difficult to do? So, like, is it the same with other lights, that there's especially, we're talking about the wavelength here, I suppose the Kelvin in normal lighting terms of a light, but there's also the wattage or the lumens of a light as well. Is that something that has to be quite precise with these devices?
Yes and no. So it's interesting. The more energy you put through it, the more that you give off, right? But it has a difficulty just because these bulbs right now need a very high voltage to really ignite this gas and cause UVC output. So the modules are very specially designed because you need to have a high voltage side and a low voltage side. But generally, the lamps themselves are 14 watt, not too much more than you would have in your normal house. I think what gets people tripped up is because we do think in lumens. You want to know how warm the light is going to be in your house, but this is a wavelength we can't see. So there's really less than 1 lumen for each of these lamps. You'll see just a little bit of a purple haze just because of the arcing in the gas in that lamp. But other than that, it's really not contributing to light in your space. So you might look at it and go, is it doing anything at all? Just because it's not a wavelength that we can see.
Interesting. You say UVC, so it has that purple, as you say, haze at the bulb itself, at the light emitter itself, but actually the light it's emitting is completely invisible. It's a dark light, isn't it?
Exactly. In that sense. And it's kind of interesting because we have dyes that are sensitive to the light. So if you're unsure whether your lamp is working, you can use one of these like little check cards, almost like you would take outside. Do you remember those t-shirts that would change color when you went into the UV light? Same idea. And you can check to make sure it's working. And of course, there's some fall off. If you've been running it for 10,000 hours, you might be around your 70% of your original output. But that's true of most incandescent bulbs. So it's nothing out of the ordinary. They actually perform quite much like you would expect them to.
That's really interesting. So that kind of answers my question in its changing characteristics over time. So because it's a gas, the wavelength will never change. It will
Right.
precisely deliver that 222. What will happen is it will lose its potency or its power over time, like all light bulbs do.
Exactly. So that's one of the important things for using this technology is proper maintenance. You can't just put in a bulb and say forever we are protected. And that is the unfortunate thing that sometimes happens when people put these devices, more traditional UVC in their HVAC and forget about them, or whoever installed it has moved on and nobody knows where to get another bulb.
I assume this technology is relatively difficult to make. This isn't a fitting we're going to be able to buy down our hardware store anytime too soon, or should do for that matter. I mean, this is, I'm assuming at the moment, this comes with a cost and is a very technologically advanced product. It's something that's an engineered solution at this stage. It isn't just something you buy off spec out of a brochure as an engineer.
No, you're right. There are only, I want to say, 5 or 6 really reputable manufacturers of far UVC lamps at the moment. Plenty of people who are trying different ways, right? So maybe it's not gas in a light bulb tube, maybe it's in an array. So people are trying slightly different things, and everything has their plus and minus. But you're right. You can buy the bulbs from these manufacturers. They're usually just component manufacturers, and divine your own lamp. So you'll see a lot of lamps, and a lot of them will use the exact same bulbs because you want something that's dependable and reliable. And there's only a couple of sources. And that comes with a cost attached to it.
So what's actually happening then at a cellular level, on an organism or on us? What is far UVC actually doing or not doing, depending on the thing that it's hitting? What was the actual action mechanism?
What's really great about far UVC and why it stands a little bit in a different space than the traditional UVC is because it acts on 2 or 3 main pathways within normal cells. And cells are kind of like this Rube Goldberg machine to begin with, right? We have made a bunch of different, very complex pathways. And if you throw a wrench into any of them, you can have some really detrimental effects in the cells. So as I mentioned, far UVC is absorbed by DNA and RNA. So this means in our cells, we're now creating kinks, little tiny binds in the DNA where now our DNA synthase, which is streaming along, trying to replicate itself or just copy individual genes that it needs, it now runs into these kinks and has to repair them first. And that's a whole nother mechanism of proteins coming together to cut the DNA, take out the part that's wrong, replace it with the right nucleotide, and then start the whole process again. And so when a cell has accumulated enough of this damage, the DNA is unusable. And so the cells will generally just enter a phase where they're no longer replicating. So this is one of the things we look for if we were to prove that our lights are working. So this is something that I get to do quite regularly, is go to a location that has installed these devices, take before swabs of some of the surfaces in their office or locker room or what have you, turn on our lamps, and then go back to those same surfaces an hour, a couple hours later, swab again, and grow these 2 cultures up and show that there was a lot of bacteria to start with, and there's much less able to grow afterwards. So we're stopping the reproduction of these organisms. But if you're thinking about viruses, they're not really reproducing themselves. They do go into cells and use cell machinery for reproduction. And if you mess up their DNA or RNA, the same problem happens. They won't make viable viruses. But because we're also acting directly on proteins, so as I mentioned, double bonds or bonds between different molecules absorb different wavelengths of light. And proteins, these bonds between proteins, really absorb very well the 222 nanometer light. So now it's starting to break apart the bonds that are holding the protein together. And proteins, a lot of time, their shape dictates their ability to do their jobs. So now not only is your DNA damaged, but like the proteins needed to repair that damage are broken, or the proteins needed to keep the structural integrity of the outside of your cell are broken. Or if you're a virus, the thing protecting your DNA or the ability to inject yourself into cells is now broken. So we're kind of fighting a war on multiple fronts. We don't really know which one's most potent, but the fact that far UVC interacts with proteins is one of the reasons that it's a little bit better than traditional UVC at things that are very protein dense. Think bacterial spores, think molds, think bigger structures, or even tiny virions, little viruses that almost everything that they are is protein. So it's very interesting if you're thinking about it that we're just kind of going after these things that are attacking us at every turn using this particular wavelength of light.
It sounds like something out of a superhero movie, where you're changing proteins and DNA, and some poor fella walks into the lab at the wrong time and now comes out as Spider-Man or something. Like, you're having really profound impacts at a DNA level, and at a protein level, and at a cellular level. Why isn't this stuff impacting us? It's targeting viruses and bacteria, but what else is it targeting when it's in the environment?
That's a great question. And as much as I'd love to be Spider-Man, it's not like radiation in that once you're in it, it's causing an issue, right? That's kind of a misnomer, maybe more true of atomic radiation. But where we have an advantage is because it has to do with that wavelength. So being a very short wavelength that has very high energy, it doesn't penetrate very well at all. In fact, a piece of Scotch tape will, if you put it over the lamp, will block everything entirely. It can get about 1 to 2 microns through most materials, and that's about the size of an E. coli cell, because E. coli is my favorite. I will use them as an example. So if you're thinking about things sitting on the surface of your skin, yes, you can get through the 0.5 micron virus, you can get through the 2 micron bacteria. But when you're looking at your skin, on average for a person, it's anywhere between 15 and 25 microns thick. And about 5 to 7 microns of that are dead skin layer. These are cells that are now anucleate. They have jettisoned the nucleus. They're not reproducing anymore. They're there as a protection against the sun. So this is why if you've recently exfoliated and you go out and go do some tanning, you're going to get a deeper burn than you had before because you really took off those layers of protection. So our far UVC light, while it's having a great impact on one cell thick things, it's not getting to the living tissue of humans. So we're getting all of this protection in these dead cell layers.
And it has to have line of sight. It's a light, so it's straight. It's, well, ish.
Yeah.
Won't go into that one. But I'll start talking about the double slit experiments and things. But it's, for example, it's not absorbed like a gas, like radon gas, and then affecting tissues internally. This thing has to physically be shining on something to have an impact. So if that thing that's in the air or on the surface is a cell, an organism, a piece of bacteria or virus, it will have an impact on that. But it's not, so there's nothing on us that is thin enough. I assume even the eyes, because I know eyes have always had to be protected by UV. I'm guessing they're even have got enough protection from a cell perspective for far UVC, have they?
You've got it. So it's both cell and protein when we're thinking about skin and eyes, right? So we have tear layers, which are thick, viscous soups of protein. And then we have, just like our skin, a couple of dead or about to be dead eye skin layers that, while the light can get to, these layers are sloughed off every week in your tears anyway. So we are protected in all the tissues that are multiplying. And you're right, it's not something that's cumulative. It affects what it can touch. Once that energy absorbs, it's not leaving behind a residue or anything else. It's gone.
Yeah, interesting. So does it affect all organisms, all single cell or very small organisms that are in the air or on surfaces in the same way? So are there resistant bacteria and resistant viruses, or more resistant, shall I say, viruses and bacteria to far UVC than others, or does it effectively any cellular type thing in that space that's getting shone with that light, it's starting to break down proteins and DNA? That's it.
Yeah, so there isn't anything that's been found impervious to far UVC light, but there is definitely a gradient. So it's easier to inactivate viruses. They're smaller, they're less component. And it's hard to say whether it's because of the DNA damage or the protein damage, because they happen at the same time. But those are the most sensitive to UVC light. And then as you go up, you get your bacteria. It seems to be there's a little bit of variation depending on how large the bacteria is, and that might just have to do with how much energy it takes to inactivate enough of these cells. And then you get to your molds, right? So these are bigger, they're used to being out in the sun, so they're a bit more resistant and they have more pathways for dealing with that kind of UV damage. So as you get a bit bigger, it takes more energy for that inactivation. And probably the most difficult are bacterial spores or mold spores, right? So these are structures that are very protein dense, usually quite large. And the idea is to survive in harsh outdoor conditions. So it takes more light, but it's not impervious.
So that's interesting. And is that kind of then the upper limit once you go above mold spores? I don't know what that would be, but I'm guessing you're moving into, I don't know,
organisms
what,
like
or
fleas.
organisms.
Yeah.
Like once you get to that, is it that's its limit really, is mold spores? Beyond that, you're not seeing any evidence of impact?
Yeah, so it's difficult because once you get out of the single-celled organisms, they're entire body systems. So maybe the fleas can't see or something is going on, but we would have no way of knowing, right? So there have been some really kind of interesting studies looking at C. elegans. So they're like a very clear nematode, really well-known model organism in microbiology because every single cell is accounted for. They know that the 4 cells it starts with will turn into exactly which ones down the line. So they can show that in this very thin, clear organism, they can get deeper damage because these things are, 2-3 microns thick to begin with. So it can damage other types of cells, but it's not the correct way to deal with that kind of problem, right? I wouldn't use a light to try to kill the weeds in my yard or the aphids on my plants, right? So it's possible, but it's not the best use of the technology.
No, it's interesting. And I was just interested because one of the questions I had was the potential of these things to create resistance within nature. When we start applying a process, and we've seen this with antibiotics, obviously with resistant organisms, that whether far UVC was effectively clearing the way for more resistant bacterias or viruses because it was killing off the weak ones, for example, that there was some kind of unintended consequence. But you were saying there's no evidence so far that anything at a single cell level really is resistant to this light. It's going to start breaking down DNA and proteins.
Yeah, so that's a really great point. We don't expect any antibiotic resistance because of the way this is working at the cellular level. Most antibiotics work in a couple different ways. They'll impede DNA synthesis, they'll mess with your cell wall, so it's hard to remain structured, or they'll go after specific pathways in the cell. These are all very targeted, whereas far UVC is a sledgehammer. It is trying everything all at the same time. And just like bleach, which hasn't found anything that was impervious to it because it works on so many different pathways at the same time, we don't anticipate anything being able to stop as many assaults all at one time, just because you need to survive in order to create a resistance.
One of the questions I had about far UVC and UVC in general was about the impact on the microbiome as we see it, because I think most people understand or have I was only talking about this with an asthma and allergy specialist on the podcast this week, actually, so Stephen Holgate, and we were talking about this fact that I think that human species recognize at some level there's a healthy level of microbiome bacteria and viruses and so on. When you have something that's non-targeted like this, is just trying to kill everything off in a space, is there a risk that we have a potential negative impact on the microbiome at some level, and therefore we would be applying this in particular settings where we genuinely don't want anything, like hospital settings, I guess, whereas the home, unless you were particularly allergic or you were vulnerable because of cancer treatment or something, generally we'd accept you want some level of microbiome in the space. So I'm assuming there are boundary conditions to these treatments of air.
Yeah, so I heard that as two kind of questions, like what are we doing about our microbiome? Is it really at risk? And then are there correct spaces to use this, and what do those look like? So I'll try to take it in two parts. The first being, you're right, we really do need a microbiome. It's really important for humans to have a very well populated skin flora for a lot of different reasons. One being is they do metabolics that we don't do ourselves. Think vitamin D that comes from bacteria on your skin that secrete it, and then we get it through our skin. We need that. But they're also like our little force fields. If you touch a surface that has been contaminated, there is a whole layer of bacteria that's already on your hands that they need to fight for resources and a space and the ability to adhere. So most of the time, when you go and wash your hands after touching something you know is gross, those new transient bacteria or viruses get washed off. That's the whole point. But what's really thoughtful about this is we don't know what a perfect microbiome looks like. We don't know how much is needed to be there. And of course, we all love hot showers. This really clears out our microbiome on our skin a lot more efficiently than shining some light on it does. So we don't know how much to recommend yet, but the fact that these are lights in places where people gather and then leave, and the fact that most people are generally covered either by their own hair or clothes most of the day, the only thing that's really going to be exposed is the same amounts of skin that we get exposed to sunlight already, which is technically a germicidal light, but much weaker. But since the sun is 50 billion times stronger than the lamp I have in my house, I imagine it's having some kind of effect on this microbiome. But this is such a new science. It's so cool. But like the first major study that looked and tried to characterize what type of bacteria are on us and where and their favorite environments was really only published in, I think it was 2013. So this is new, and we're doing all we can to protect our microbiome, but we don't know what good looks like quite yet.
And it's the same indoors. It's the same in the built environment. We really don't know what a healthy microbiome looks like outside. We don't know what a healthy microbiome is inside our buildings and on us.
Right.
It's a really interesting area, and until we have that level, and that's probably the big frontier of research, I think, is to try and characterize and map out what these microbiomes look like. The risk, I suppose, with something like UVC of any description is just using it in all sorts of spaces might have unintended consequences. If there is such a thing as a healthy microbiome in a bedroom or a kitchen or spaces that we live and occupy where we are touching and exposed to stuff all the time, we just don't know, because we don't know what that is. But I'm guessing in answer to the second part of the question, there are environments where we really do want to keep the stuff that's alive and floating around in our space to an absolute minimum, regardless.
Yes, that's it. Exactly. It has more to do with personal risk to us, right? We want to be able to go into shared spaces, be it hospitals, be it college, high school, grocery stores, these places where we know we're going to gather that are vital to human success, but where we don't want to go and be afraid of picking up something from the person next to us. These are places where far UVC excels. The idea that we could treat the transient pathogens that'll be in our air effectively, because really the highest risk to us and our health come from the other people in the room. It is really hard for bacteria that are meant to live in people to make them sick. Same with viruses, to survive in not their favorite environment. They don't want to be stuck to a surface. Eventually, they'll be wiped up by a Clorox wipe, or the UV from the sun will inactivate them, and that's the end of their road. So the biggest risks are going from perfect environment to perfect environment, the next person. And if we can inactivate those bacteria and viruses while they're in the air, while they're trying to transition, we can really lower risk to our own personal health. And that's kind of the space that we see this moving into, because shared air is a shared resource, but it's also a shared risk for everybody who's in that room.
Yeah, really interesting. Does far UVC have any air chemistry impacts? Like, is it like ozone? Can it create hydroxyls and radicals and do things to other compounds in a space that are unintentional? What's the evidence around UVC and far UVC when it comes to its interaction with air chemistry in a space?
Sure. This has kind of been the new and exciting topic the last 5 or so years. So there's lots of papers that have come out and looked at far UVC in small chambers to really try to see exactly what chemicals are present and which are changing. And what they found really is the biggest change comes from ozone. So 220, these bulbs have a little bit of light that comes out near that 200, and that's where you get the most ozone generation. And I say most, but still very low. So, for example, the lamps that we work with, one is 0 ozone and one is 7 parts per billion, where the OSHA recommendation of limits in a space is 50. So that 50 parts per billion. This is tiny amounts of ozone generation. But just like the ozone that comes in from outside, it does have an effect. So if you have a lot of hairspray or these scent compounds that are present in our spaces, they tend to degrade into lower or more reduced components. What we've seen is most of the published papers focuses on limonene. So this is a kind of a lemony, pine solly smell that can come out of some cleansers, and they see that chemical is reduced in these environments. And so it does give you this chance for particles to build up in spaces, but that's why we really, really want people to focus on putting this technology in spaces with proper ventilation already. These things are only something that can build up if you're not changing out air as you're supposed to. So in fact, we recommend having at least 2 air changes in spaces, and don't install when it's below that, because we don't want to put any risk in these spaces. We're trying to limit risk, and we don't want to add anything that could be a problem. But most of the publications that have been out there agree that these changes are very small and really only have a build-up effect when you're not properly ventilating a space.
And there's no such thing as zero risk. You know, we can't exchange one risk for zero risk. It's not the way the world works. You know, we have to look at what's important to us, and if there's an infection from viruses and bacteria, or the potential of a very small amount of ozone production, that may be a risk that is something that people are willing to consider. I think often the interesting thing with the studies, and I know speaking to people in academia, the focus is on the level of ozone production rather than the changing chemistry in the space, because that's much harder to measure. Right. I know one of the interviews I did was with John Wenger down in University College Cork, and they've been studying free hydroxyl radicals in the air, and really you're talking about parts per trillion of these radicals, but they are fundamentally the engine room of air chemistry because they're so reactive. They are driving so much of the basic chemistry that we see in our environment. So it's incredibly difficult to measure. But I suppose that's just something that we just have to keep looking into and seeing what, like you say, if you're getting a decent amount of air change rate, that's the thing that stops ozone building up and those other chemicals building up in the first instance.
You're right. It's not a zero risk game. And I think where we really try to focus and understand is kind of that risk benefit analysis. Now, when we're having good ventilation in a space, your room will start mimicking the amount of ozone that is in your outdoor air, right? Because you're bringing it in from outside. So on an average day, it's usually 30 parts per billion outside, unless you're in Colorado where you're on top of a mountain and it gets closer to 60 parts per billion. And if you're bringing it inside to get rid of the particles and gases like CO2 that build up indoors, you're just changing one chemistry for another. So it is a balancing act. So I don't want to argue that it's not doing anything to the chemistry, but it is. But I'm not saying that it's bad.
Charles Weschler was saying as well, it's not even as simple as the amount of air change rates, because some chemistry happens faster than others. So if you're moving air very quickly, you only allow the really reactive chemistry to be present, which might be good or bad. It just depends on what that chemistry is. So this stuff's really complex. Even if you have an understanding of what's going on, it's very difficult to control. Absolutely, that you're going to be bringing in some of these things. So we're not seeing major air chemistry changes effectively from these lights in the spaces, from what we see.
That's right. So for most spaces, when they're using it as instructed with these lower fluence values that really target these bacteria, we're not seeing a buildup of any type of gas, hydroxyl radical, or reduced product that is causing for alarm. So this continues to be studied and is something that we always get to talk to at the International Far UVC conference that we had this year in Boulder, Colorado. So we got to be in the high zone ozone environment and get to talk about it. It was great.
I'll have you back to the podcast in just a moment. I just wanted to very briefly tell you about Farmwood. Farmwood is one of the most successful SME ventilation companies in the UK. Multi-award winning for their work in ventilation, compliance and health and safety. Farmwood has spent 23 years becoming the go-to service partner for a range of UK and EU ventilation manufacturers. They work on commercial, industrial and residential projects, and they're all about linking up academics, industry and engineers, driving real regulation reform, and most importantly, making sure the buildings we live and work in are healthier and safer for everyone. Their managing director, Nathan Wood, is a fellow campaigner of mine and someone I respect deeply. He is the chairman of World Ventilate Day and was previously the indoor air quality chairman at BESA. He's now leading the charge for competence and compliance across the ventilation sector in the UK. Farmwood are also really big advocates for apprenticeships, helping to develop the next generation of HVAC engineers who are going to keep our buildings breathing. They're a company that genuinely cares about buildings and people. You'll find more details in the show notes and at farmwood.co.uk. Now, back to the podcast.
Are there any co-benefits to using far UVC? So we've talked about its impact on organisms effectively, but does it do anything else? Like, is it breaking down VOCs or doing anything else at any other level, or is this really a very specific thing that far UVC does? There aren't other co-benefits that we engineer with it?
So I've seen a couple of reports for studies that have started to look at other things besides just pathogens and gas. So one study looked at the ammonia smell you get from urine being in a room, and they show that it reduced that smell of ammonia in the air, which could be really interesting in a bathroom or hospital setting. And I got to see one presentation, and I'm still waiting for the paper, who were looking at the proteins that cause allergic reactions, be it cat dander, pollen, that kind of thing. And we're looking at the protein components, putting it in the air and then turning on a far UVC light, and then recovered the protein from the air again. And they saw that because this acts directly on protein, they've recovered less of the allergen than they originally had. And then by doing an immunoblot, they showed that it seemed like there was less there. So it's possible that this light is able to reduce the effect of these allergens, and maybe to how they affect humans. That part needs to be done yet. We know it reduces the amount of protein. Does that mean lower reactions for people? Not sure yet, but I'm very excited for that science, and I will keep watching that lab with great interest.
Yeah, and I'd imagine a lot of the population would be interested in anything that reduces pollen, particularly.
100%.
Dan, yeah. Yeah, really. But that again is another size up from even mold spores, isn't it? Pollen. I mean, pollen, you're starting to get quite complex
Yes,
things.
exactly.
Yeah. Really interesting. So what does this actually practically look like on the ground? Like, so where is this stuff going, and what does it actually look like? Are we just replacing GU10 light fittings in ceiling tiles, in a hospital room? Like, practically, what is far UVC manifesting as in what spaces? Because I think people have understood the UVC technology of the past, where we've had to shine it up at ceilings and keep it away from spaces and so on. Practically, what is far UVC actually manifesting as in the spaces where it's doing good?
Sure. So far UVC, because we know that it can be shined on people and put in occupied spaces, the form factor looks much different from the tower type traditional UVC applications. So usually these devices are installed directly in the ceiling to shine down in the space, or sometimes they're put on a stand and put in a corner to shine long ways, to get a really long path length that have an effect all the way down. But these can be as simple as replacing a can light in your ceiling. So we do have to keep in mind, it doesn't add any lumens, so you can't just take out all your lamps and put in fire UVC lamps. It will be a very dark room, but it's almost something that you don't notice anymore unless you're looking for it. Unless you look up and you say, hey, that lamp is slightly purple. What's that? So it's really interesting in the places that we've been able to put it, because it's so inobtrusive and it's not something you need to haul into a room and then take back out again. So we're seeing it in academic spaces. We put it in a lot of different lecture halls. Of course, it goes in bathrooms and hospitals, places where, infection risk is really front of mind. But it's been going into places that maybe we weren't really thinking about. It's going into athletic teams, people who want to protect their staff at all costs, because that's where the important part of their company is. Think about teachers. It's gone into a couple of early child learning facilities, because these are where we need to be most robust. And we know that things kind of hop from one person to another very quickly. So places where the staff are really important, where there's a known risk, or whether there's somebody just passionate enough about it, right? So we've had the wonderful pleasure of working with the LECOM Medical School, and it really started with a discussion with their chair, who's just so excited about new technology to fight germs, being a bit of a germaphobe themselves. So it's interesting to see how these get deployed, but you might not even know they're there. It's worth looking around.
And is this engineered into a space? So a bit like good lighting design, are there certain angles of light that you need, and a certain amount in a coverage in a certain space, and depending on the height of the ceiling? And like, is this a technical process to place them, or can you just chuck a couple of lamps in a space and it gives a good wide? How does it work practically when these things are going in? Somebody have to come out and figure out how many and where these things need to go?
So to do anything correctly is technical. It's not like you're just providing light to a space, and if it's not dark anymore, you've done it right. We have objects in our space. There are things that'll cause shadows. And as we already talked about, if the light doesn't reach that surface and that was your favorite surface and that's what you want cleaned, it's not going to work. So we do have guidelines for us internally for how many we put in a space. Depending on the room and what you're hoping to do with it, we usually aim for about one device for every 200 square feet. It goes to about 150 if you're in a medical facility, just so that we have the faster turnover of the bacteria in the air. But it's not so hard that it couldn't be done. It is looking at your space critically, thinking about where you really need the light, and making sure that you have it where it's not getting shaded. We've had the misfortune of sometimes we thought it was fairly straightforward, and we gave them a plan for where it should go in their ceiling, and they thought, well, it would be easier to put it closer to the wall. And so they did. They put it directly next to the wall, but that blocks half of the light that you were going to be putting into that room. So it's just a little bit of basic understanding of light in a space. And as long as you're not shadowing, you'll be able to have a good effect.
Does the environment impact its effectiveness? So things like humidity or particulate matter in a space, and as such, do different spaces require more or less of them to be effective?
Yeah, so humidity and the amount of stuff in your air does make a big change. Usually, if it's a really moist room, it just doesn't work as well, because now not only is the light trying to get through the space, it has to get through these droplets of water. And as I mentioned, the energy is really easily absorbed, and that could be just enough where you might be, 60% the amount of light you thought was in that space is actually there. So we do recommend it for areas that have ventilation and is not really humid. It's not best for like a shower room at a gym. Unless they have good ventilation to go along with it. For the particulates, that's a harder one, because if you have an HVAC system and you have particulates in the room, be it from smoke or from animals, as we start to put these in barns, there's not a whole lot you can do to change the particulates in your spaces except for adding more filters. So it's just something we have to think about, put more lamps in the space just to make sure that we're getting through that cloud that you're working in.
Yeah, interesting. So what are the kind of environments that you're finding this tech going into then? What are I think you said animals just there as well. Is this going into chicken houses and animal husbandry facilities and places where you're trying to sanitize and keep stock healthy as well as human environments?
Yeah, so animals are a fascinating place for these technologies to go. It's kind of a newer space. There's a couple of companies that have been doing it a little bit more regularly in the past couple of years, and we're doing some pilot studies because we really want to know the effect and what we can promise as reduction of pathogens in these spaces that have a lot of things going on, right? They have animals that don't necessarily bathe themselves every day. They've got air ventilation where it generally just comes directly from outside, maybe goes through a filter and then goes into these spaces. So it's not ventilation like you and I are familiar with, where we have a better chance for particles falling out of the air. So we're doing a study in a pig barn in order to understand, can we reduce the amount of viruses indoors, and does that change the amount of viruses shed outside? Because the idea here is, if there's a problem in one barn, these barns are right next to each other, you could be just passing it to the one next door. So this is a really interesting environment where biosecurity is already something they spend so much time and attention to. I don't think people realize just how much thought goes into a lot of these facilities. So they're always looking.
There are more air quality standards in food production than there are in our own houses and workplaces. I mean, that's reality.
It exactly.
Yeah. There's a lot of money at stake. I mean, you can wipe through an entire chicken house with poor ventilation and viruses and bacteria if you're not careful. So, they do take this stuff seriously, and there's a lot of money on the line, of course, as well.
Right.
This is production.
Yes, but it's always good to have the data to back it up. So just saying that your neighbor's barn did it might be a good enough for one person, but to really get into the industry, we need some great research. So we're trying to build a partnership with a couple of universities out in Iowa and Minnesota in order to do a study looking at the barn specifics.
So back to our world then, where are we starting to see far UVC getting some traction?
So definitely in the medical spaces. So we've had a lot of installs in areas where specifically immunocompromised people are going for appointments, which has been really good to see, because there are more people who are immunocompromised than we really give a lot of credence to, and they still have to interact with our world. They just have to do it at a higher risk. So the fact that hospitals like Mount Sinai and different clinics are able to start building in extra protections for these individuals, I feel really great about. And that's where we've seen some movement. Additionally, places where people are gathering. So like I said, teaching universities have taken this on, places where we have a lot of bioaerosols. So there's more uptake in dental clinics than you would really give first mind to, but it should be in more places. It should be in the ER where you don't know what someone is coming in with. Like, these are places where there are high risk, but it just takes longer for the installs to go through. We have to get approval with a lot more levels within a hospital to be put in an ER. So fingers crossed, it's one that we can move into soon.
I know with UVC, where we've typically treated it in ducting, and like you say, where it's out out of human sight to traditional UVC to impact air quality. There's a kind of a linger time required for air in the light to be able to be a certain percentage of effectiveness. What's the effectiveness of far UVC in a space? Is there a point at which the air change rate overwhelms the ability for far UVC to be able to have an impact on the air it comes into contact with, or the viruses or bacteria that it comes into contact with? Or is it almost instant the moment a bacteria or a virus has that light on it, it's damaged, that's it, it's done in an instant? Or does it need to dwell in the space for a certain amount of time to have an impact?
So these are one of the difficult questions, right? It all has to do with the amount of light this organism absorbs and that time factor, and that's part of that dosing equation. So if you're putting a UVC, a far UVC lamp in your room, what's different from traditional UVC, instead of just pointing at the ceiling or being in a duct where you have a very small volume of air that you're treating, you're now treating the whole room always. So now your whole room is your duct. So we have a lot longer dwell time than you might in some of those other spaces, which works to our benefit. But of course, we're now working with air that's mixing. So we're getting kind of an unknown value for each of these individual pathogens. But what we can figure out is that if a specific pathogen were in this light, how long would it be for 50% of them to be inactivated? And the order of magnitude for that is actually really quick. So like H1N1, we're thinking flu, these viruses in the light, depending on its distance from the light. But if I even use a room average, it might be a minute, and now I've gotten 50% of that virus inactivated. And it gets a little bit longer for like a Staph aureus, where it'll be closer to 3.5 minutes for 50% to be inactivated. But 50% can be everything when you're thinking about risk, because it doesn't need to be 99.9% for this to have a huge impact on the rate of transmission between one person to another. So this is one of these fun but difficult math problems that functional biologists and people who do quantitative studies can look at, using the Wells-Riley as an example, to see if you're looking at production of this many quanta per hour and the virus or bacteria is this susceptible and the people are this active in the room, you can actually get a good estimation for what type of risk reduction you're getting in that space. And this is one of the things we actually partnered with the University of Arizona to do a study with our lights using their simulations. And we're seeing that if you do this in like a room where you're working with someone for an hour, you can reduce risk by just turning on far UVC in this space by 91% for an hour. That it's mind-boggling, because this is where people get sick. It's going to the office and sitting next to the coughing person. 91% reduction in risk is huge. This is now putting it under the acceptable risk, is something that's talked about in this field. The idea that 1 in 10,000 people in this space would pick up something from the sick person in this space. That means, 0.01%. Whereas now in these spaces, when we introduce the lights, we're getting close to that acceptable risk value just by turning on a lamp. And I think that's where the strength is people have to choose to wash their hands, to protect themselves in other ways, like with masks or vaccinations. But if you just clean the air, we could lower risk for everybody in the space.
Yeah, I love that analogy with clean air. It's a favourite one of Joseph Allen's, I think, is if you have good air quality, all you ask of people is that they sit in the space and breathe to reap the benefits and rewards of it. It's a very low effort impact on performance and health and well-being is if you can create good environments, all you ask people to do is turn up and breathe the air that they're in, whereas all these other things require so much effort. Right. It's almost incomparable. It's a beautiful segue actually into the kind of the second big question, which was, and we've started to talk about outcomes here a little bit. And this is what's interesting, I think, with far UVC, is it's starting to sit in this new world, particularly in the States with standards like 241 from ASHRAE, where we're starting to think in terms of equivalent air change rates, that actually we can derive an outcome in a space not just by replacing the entire volume of the air in that space so many times an hour, but we can treat it in different ways, whether it's filtration or with light or with whatever it is. There are now equivalences to that air change rate. Where's far UVC sitting in that world right now in being able to quantify its impact on a space and convert that into metrics where engineers can start to say, well, look, I need an equivalent air change rate in this space of 7 air changes an hour. It's a classroom. Or that's my target rate. I think I can achieve 5 air changes an hour with the traditional HVAC system, but I want to achieve an equivalent air change rate of 3 with other measures. Is far UV starting to sit in that picture of spaces where we're taking part of the role of traditional air replacement and saying, actually, we're going to contribute this? Or does it just sit on top of everything and just add an extra layer of protection? Or is it becoming a component part of that air change rate, do you think?
I think it's going to move to be more of a component that we're thinking about continuously, because right now HVAC systems are kind of at their limit. People don't want to put more burden on them because it is costly to turn them up to bring in more fresh air from outside, and it's costly to replace them. So what can we do besides overtax this technology that we already have to get more clean air? And that's one of the places where UVC and far UVC shine, because it is low energy. It's usually something that's autonomous. You put it either in your duct or your room, and it will just keep providing that clean air. And you're right, there is a fabulous math equations that really can tell you about the amount of reduction of pathogen and equate it just like they do for reduction of particles in these spaces. And that's kind of right now how it's bridging the gap, is understanding, like, if I was to just run the HVAC at a high enough to create the same reduction of bacteria, that would be the equivalent air exchange rate, the equivalent clean air rate. So that's kind of how it's sitting in the standard right now. But what's really powerful is because we're starting to learn more and more about how susceptible viruses and bacteria are to this light, that we can see in normal spaces, in a room my size, which is about 10 by 10, which is kind of a small office, but we could have a reduction by about 13 air changes equivalency per hour by putting one lamp in this room. The difficulty comes from setting some standard for that, because that number changes depending on what bacteria or virus you're looking at. It's a calculation based on their susceptibility. So we, as individuals and experts in the field, can work with ASHRAE to find out what is the best proximal that we can use. Because it used to be, for filters, they used MS2, which is a bacteriophage, so a type of virus that infects just E. coli, and used that as a proxy for all of the other pathogens, because it was a small enough particle size that it was very difficult to get through the filters. But it's not the perfect replacement to use for UVC, because it is this particular shape of virus, it's just really hard to inactivate using far UVC light. So it's not the most difficult thing for the filter, but it's the most difficult thing for us. So we're trying to find something that can be used across different platforms in order to give the best estimation of what's a reduction for one technology and how it applies to another, which is exactly what ASHRAE 241 is starting to do. There's an entire supplementary table where you can go in and calculate, okay, if I have this type of filter and this many air changes or cubic feet of air brought in per hour, how many more do I need? But far UVC and UVC is over and above, based on energy, able to provide clean air. Actually, the Department of Energy did a study in partnership with the Pacific Northwest Labs PNNL, they did a great study looking at if you just turned up components of your HVAC as high as they could you reach that new CDC recommendation of 5 air changes per hour? And the only ones that could do it were in-duct UV systems, in-room UV systems, and far UVC without really breaking the bank on energy. So it's something that we need to start considering, but it's a slow adoption so far.
Yeah, you kind of need a testing methodology, a bit like CADR ratings, where you start to have a range of bacteria and viruses as the metric, as opposed, to something that's fair or across the spectrum to everything from filters to, I don't know, what you call it, ionization technologies to UVC and far UVC and so on. And so that there's some a fair test, basically, that can replicate, a bit like with CADR, where you have different levels of particulates and smoke to see how efficient a filter is.
Exactly.
You could have different types of viruses to measure it. I mean, the thing we're talking about here, interestingly, is infection risk and protection, which is where 241 is centered around. But I think the interesting thing about 241 is that it's starting to open up this idea that actually equivalence in air change rate is something that we can consider for air quality in general, not just infection risk. But the challenge there is that infection risk is just one ingredient of the cake of good air quality that you're trying to reach at the end of it. And there it becomes quite complex, because it takes quite some level of expertise to say, I'm going to deal with this element with far UVC, I'm going to have to do this much with filtration, I'm going to have to do this much with gas phase air cleaners. If you're trying to get an outcome equivalent to good air quality, you're not just going to achieve that with far UVC. It's going to be a mix of other measures that would have to be included. And at the moment, I don't know where that expertise lies. That's a complex mix of engineering, air chemistry, and infection control, isn't it?
It really is. I think that's one of the strengths of these standards, is because it is trying to attest to the gases in these spaces, the particulates in the spaces, the chemical exhaust that might be happening, your outdoor air, and pathogens. It's a huge standard. And the reason why we have so many different people working on it, because you're going to assess one thing at a time, decide how to remediate for that one thing. And it shows that HVAC systems, that clean air in your indoor environments, really are a whole network of things that are working together to make safer air for the people indoors. So it's not just one solution. UVC doesn't address with the CO2 buildup in spaces. HVAC does some things with viruses, but it's not enough to really reduce things unless you turned it up higher. And our filters can't push or pull air, so we still need HVAC. It's all about creating this perfect environment, and we all have to work together for that one.
What are we not talking about that we should be when it comes to far UVC at the moment? What are the things that we should be focusing on, or researching, or innovating and putting resources towards with far UVC that we're not at the moment?
That's a tough question. So right now, the technology is young enough where we're still asking the questions like, well, how is it applied well in this space? What about this, my favorite pathogen? We're kind of still feeling out the edges of these things. And the chemistry is one example of the thing that we're just starting to get a deep dive into. But what we really need to show is how this changes day-to-day things. We need a long-term human study to show that if you put it in a space, the people in these spaces, these people in these offices, have fewer instances of respiratory infection. But that's something that takes time. I know there's been a study up in Canada who's doing a double blind in a long-term care facility and tracking respiratory infections, and it's been going on 3 years, and it's supposed to be done this fall, but that will be a 3-year study. But in our first look for how much this actually impacts us longitudinally for illness. So right now we can talk about, yes, it works on all pathogens, yes, it makes cleaner air, but we can't tell you what that means to our health, 50 years from now. Does that mean we were healthier? So I think that's the space where we need to move into, because people care when it changes their personal risk. And I don't think we have enough funding for that.
You could have literally been in a room with Mildred Wells, 80 years ago, having the same conversation. It's that we don't have the epidemiological evidence that we need to say these are the spaces that this stuff needs to be applied on, because this is the public health impact of using this technology versus not using this technology. We're still at the chamber research and potentially use case, like project case, like a care facility end of it. We still seem to be a long way away from the big evidence, the big data evidence, where we can say, actually, these cohorts have been exposed to this technology predominantly through childcare or through school or in elderly care, and these are the impacts with that cohort at the end of it versus those that weren't.
That's it, exactly.
That's where we need to get to.
Because then we can make the case for why it needs to be everywhere. Right now, yeah, you've solved my problem when I'm at the gym or the office, but I go on a bus to get home. I go to the grocery store. I kiss my kid goodnight when I get home. I can no longer tell where I got sick. So we need to really do something similar to what Wells did, where you kind of pick one town and make it the cleanest town in the world and then compare it to its neighbors. And until we do something like that, I think there will be enough reticence for a new technology where, unless you're really passionate about it, you're not going to make a change unless somebody else makes you. It's hard for people to do something like use an invisible light to kill an invisible organism that, if it works, nothing changes.
And funnily enough, that's what, ironically, the Welleses were trying to do back in the day. Was they had a few local mayors, I think it was, or councillors that decided to put these in schools and hospital facilities, and they were starting that in the very early days with, I can't remember whether it was TB or something like that, but it was trying to get there. You know, we need that kind of large scale district level almost deployment of the technology to say, look, when this particular flu swept through society in 2028, this town was affected much less by it because people were exposed to these types of lights. But like you say, you're trying to convince people to put these invisible lights. Everybody needs a monorail, according to The Simpsons, you know.
Yes, you're right. And Wells was doing it for measles, which was highly contagious.
Measles, that's right.
Things that have higher risk and known risk, things that are endemic, they have used this technology. So you'll think there's a lot of studies that came out of homeless shelters specifically because they used upper room UV to deal with tuberculosis, which was a known problem in these types of facilities. And they continue. It's actually one of the biggest prevention measures for tuberculosis in the developing world as well, because the technology is inexpensive and it can be used. So we don't, on a day-to-day basis, think about the illnesses that we encounter in a year as something that's avoidable. You don't think you can get through flu season without getting something, or at least stuffed up a couple of days. And until we believe we can change that, I'm not sure there's enough self-interest to do something. So I think it's going to have to be somebody who's just ready to be a pioneer like Wells was.
Yeah, I know. And the conversations like this always remind me of, there was this beautiful image that came out during COVID, and it was at a time where they were trying to look at social distancing and they were looking at fomite transmission. It was the very early days of trying to reduce the risk of infection from COVID. And there was this picture of outside of a primary school of there were signs on the outside of the school about social distancing and wearing masks, and there's a picture of a kid licking a railing.
Oh no, but that's it.
And you just, like, you know, there are some environments that are clearly higher risk than others, and you know, you send your kids to the Petri dish, which is preschool and primary schools. You know, it's kind of why you send them there, almost, is to build up a tolerance to all these colds and flus. Most parents know you spend the first kind of 8 years of your kid's life constantly with a cold and flu because they go into these environments and bring them all back. But there is this assumption that it's an unavoidable risk.
Right.
and I think that's an assumption that we can break somehow.
One of the things I find so interesting is just how recent this taking on of our own personal public health happens. Like, we didn't have clean drinking water as a bill until I think it was 1974. And we have a Clean Air Act from the 60s, but that's just for outdoor air. Can you imagine if we standardized and required clean indoor air, buildings to provide pathogen reduced air? It's something we can ask for because we've asked our government for it decades and decades before now. But right now, no one takes responsibility for the illnesses you get in the office because they'll tell you you got it outside or somewhere else or from your kids. So unless the government mandates it, I don't think we're going to have people taking responsibility for the fact that you could have less illnesses if we just made sure all indoor air had reduced pathogens.
From a pathogen perspective, do you think we can get to a point where we can actually set a standard, a threshold for what you find in a space, like formaldehyde or PM 2.5, that there's a culture threshold or something? You go into spaces all the time. You go into hospital settings and clinical settings and non-clinical settings. You said at the beginning that you swab before and after. Do you think you can get to a point where you can get a target value in these spaces that become a standard that you hold people accountable for?
So it probably won't be done in the same way it is for those other type of chemicals, just because the sources are the people that go into these spaces. So for a hospital, yes, we can make sure their clean room is verified. There are, in fact, levels of how clean things are based on how many bacteria are on a square centimeter. And if they're above that, they have to close down the room and clean again. But those are on surfaces. And it's specifically because air is so transient. My problem doing these before and after tests is if I go in at the very beginning of the day, the air would be very clean because there's nobody there breathing and coughing into it. So you actually have to think about how many people are in that space, how many particles they might be shedding. So instead of looking at it from that angle, there will probably be a standard that could be made based on how many equivalent clean air changes should be provided to that space in order to make sure that if it was based on a particulate size, like how many particles are allowed to be here, we can reduce it to X% within an hour. That's probably how it'll do, because otherwise you're now chasing things that are generated every other second by people breathing, coughing, sneezing, touching things, which isn't the case for other gases, except for CO2, which happens at a very standard rate because we all need a certain amount of air to live, whereas pathogens don't play by that same rule.
This is all very, very specific. I'm fascinated to understand how you ended up here, Janet. It's an odd job to have landed in. What was your kind of academic and career path into kind of technical roles in a light bulb company, to make it sound as simple as possible?
No, I love it. I have had kind of a strange path, well, at least from my perspective, because I kind of started this as being someone who loved science and was a bit of a germaphobe. And what did I do? I got a PhD in molecular biology, which focused on E. coli labs. So I was looking at bacterial host interactions and understanding their role in both our environment, in biofilms, but also their impact on our health. So this is where I really started to be interested, because I was able to ask really specific questions like, how much do we need to reduce a biofilm so that we have a health benefit? So I worked with labs that did things with dental plaques and biofilms on contact lenses, which are gross. So don't read those papers. They make you a germaphobe if you weren't one to begin with. But I finished that degree at a very interesting time of everybody's life, at the very beginning of the pandemic. So what I had been doing in this microbiological lab, where I knew I wanted to get into industry, was volunteering my time. I love science communication. So I was doing things with the local Natural History Museum, giving talks to whatever 5-year-old held my eye contact a little bit too long, or whatever adult was just wandered and bored. But in addition to that, I was volunteering my time with some of the local founders here. So I'm in Ann Arbor, Michigan, at the University of Michigan, where I did my PhD. But we have a great biotech scene. And there was one person who was trying to start a company that was making a hand washing device using far UVC light. And they just needed some proof of concept things. So I swabbed some plates, I swabbed a bunch of hands, and then everything shut down. So here I am doing something that's important and relevant to my day-to-day life, going around begging people to let me swab their hands before and after they washed them with and without these lights in order to show that there's an impact. So I immediately felt like I could start making a change in this space. And I really fell in love with the idea of asking less of people, but still having a great outcome. So far UVC really seemed like a great solution that's just so underutilized. And I didn't know if it was because not enough people are talking about it. It's just not funded very well. And I thought I could make an impact. So here I am, 5 years on, still having worked in the UVC space and just keep getting these opportunities to talk with wonderful people and work with people who really want to do good. I've actually gotten involved with ASHRAE myself so that I can make sure that we're creating standards that can build this into it so people can do anything to help reduce risk to people in spaces and talk more about it. Because I think that's the problem sometimes with highly technical new things, is that it seems too much to take on, but really reduction of pathogens in air means reduce risk to people. And that's a pretty easy in-out thing that people can understand.
Yeah. And, we overcomplicate air quality. And to be fair, some of it's very difficult. And there are elements of the impacts of air quality on human health, for example, particulate matter, that are very complex and multi-systemic. They're both toxicological and epidemiological in nature and research, and it's very difficult to draw straight lines from a particular exposure risk to a particular health outcome. But a bit like radon, viruses and bacteria is a bit of a slam dunk in some ways, because we know a certain virus is going to give you a certain health outcome, right? It's a very straight line. And if you can do something, if you've got a known engineering control that can reduce that risk, you can draw a straight line to a particular reduction to that risk. Now, you can't control every space that somebody's in, and they might have got sick at home or on the bus and so on, all those usual confounding factors that exist elsewhere with air quality. But at the very least, with viruses and bacteria, it's a straight line between exposure to that thing and an illness or an impact on your wellness to some degree. So it should be much more prominent than it is. But we're in a funny space, and I don't know if you get that in this weird post-pandemic twilight zone that we seem to have found ourselves in, how quickly we seem to be wanting to forget the lessons 5 years ago that we all hard learned. And you're at the coalface now in industry, aren't you? Like trying to help an organization get this tech. You survive by getting this tech into spaces.
There's definitely some COVID fatigue. People don't want to talk about the fact that there could be something else coming up. It was a bad time for everybody. We don't dwell on it. Not only that, the funding that was present in COVID has gone away. So people who have picked improvements and maybe they weren't the correct solution, or it was deployed incorrectly, or maybe they invested in some of those wands that you really had to hold there for a whole minute if you read the instructions, right? So people felt like they stick their neck out for some technology and got burned. And I understand that that is a really hard thing to overcome, which is why I think we spend so much time on the science communication and improving ourselves, doing research, partnering with people who are doing the research, or taking swaps myself, because they're no longer going to be like, I trust you. I've heard this name before. Show me how you've worked it in a space and then I'll believe you. So that's kind of what we're running up to now. But I'm happy for the challenge, because if a technology truly works, it shouldn't be a hard thing to prove a second time, right?
So what does a good trajectory look like for far UVC from here, do you think? What are the kind of building blocks we need to start to see put in place for this start to move from a very early adopter pioneering stage to more mass adoption of the technology and the kind of environments it needs to go into?
So I think we've done some of the early steps already. We've got great scientists who are putting out papers every year showing in the newest configuration in this particular room with this weird virus, it has an effect. So the question of, does it work? Is it safe? What about the emerging things? That doesn't seem to be the first thing people ask anymore, which is great. The second step was standards. Having something that is part of ASHRAE or building standards or government standards or testing standards really gives a lot of credibility and kind of guardrails for the field, right? You're not going to have a device that's gotten UL certified and then not put out the energy it promised, like those tiny wands lied to some people, right? So now we have a way to make sure that the devices that are getting out there are good and are doing the things that they say they are. So the next step is really getting people to start to advocate for it, right? Everybody is taking health in their own hands, and they're finding things in spaces in their public sphere where they can make a difference. And I know a lot of people I've talked to who would love to put this technology in like their care facility or their YMCA or something similar to that. Right now, it's a funding thing because the lamps themselves are a little expensive and there's no longer most grants for them. The places that need them don't generally have the money to fund them. So we need someone to be this champion. We need a politician to stand up and say, we're going to make a grant for improvement of indoor air quality. We need someone locally to say, we're going to have the cleanest subways in the world and put some lamps in. We need someone to complain to their office that Karen from finance keeps getting them sick and holding people accountable. And once enough people show up, I think the change is going to happen really quickly because the technology has gone through the hoops already and has the guardrails there in place.
So on that standards front, I think there's a really good layout of the trajectory actually. On that standards front, today, if I go out to buy a lamp or a solution in far UVC, is there a standard internationally that bulb can be held accountable to, or an outcome can be held accountable to?
So there are a couple, and it differs by country, which is a little difficult. So, for example, Europe has a lower amount of light where they allow you to put into a room, whereas the US has a higher value based on the ACGIH, the American Conference for Industrial Governmental Hygienists, has set this value. Thank you. It took me a second. But they set this value based on looking at exposure testing, saying people expose this light onto skin. When did they start having any negative effects? And they have changed that value because of all the new studies that have come out in the last 30 years. Europe is doing a wait and see thing to see how the US reacts to this change and what these devices will do. So I think having that long-term data will help both of us country-wise. But you can get devices that have been UL certified as a lamp. So not only is it outputting the amount of light, but you can go through the UL 8802 process that is specific for UVC devices. And now you can certify that it is safe or risk exempt for occupied spaces, which is something that didn't exist 3 years ago. So you can look at a box and now see if it fits some of these standards and is performing and putting out as much light as you had hoped. Whereas before, you just said that there was 2 LEDs and we're like, yes, it does something.
So importantly, it can be procured and specified. That's the main thing, that somebody can put an equal or approved qualification to the specification of the light bulb and to say this is what we expect it to do. And is that standard, is there a standard specifically for the far UVC element of this, not just UVC in general? So there's specifically something that says far UVC at 222, this is the amount of light that it needs to be able to give out or shouldn't go above or whatever it is.
Yeah, so ACGIH sets that limit. It's different by every single wavelength. So when you do that 8802 testing, they actually take a spectra. They measure the amount of light at every individual wavelength, use a very long equation to measure the amount of risk for each of that wavelengths, and making sure that if the cumulative risk doesn't come over a certain value. So if you fall under that, you'll be risk exempt. You can also be category 1, 2, and 3. Then it'll limit how much time you can spend in these lights. So a risk exempt in an 8-hour day for a work day, which is what these standards are written to, you can be in it for 8 hours. There's no limitation on time. And as you step up, it's 3 hours, it's 1 hour, it's like 30 seconds for the very high stuff. But that's more for sanitizing devices instead of people.
From what you were saying earlier on, is it zero risk being in far UVC light, or is there a limit? Is there at some level a risk to a human being in far UVC light? Why couldn't you be in it in a hospital environment, for example, 24 hours a day for 7 days of the week over a 3-week treatment period?
I will say that the ACGIH limit is not the line between safe and unsafe. It is the limit we're setting for what we know is safe right now, not saying that it's not above that. Evidenced by the fact that they did change it in 2022. So when I say risk exempt in the UL standard, that means it's below that threshold. Not that that's when you start getting the effects. So all of our devices for our company, you're able to be in 24 hours a day as long as it's mounted in the height that it's supposed to be, and you'll be completely fine. And there are studies that have looked at exposures over and above that ACGIH limit. I think one of them came out of Leeds, and it was 37 times the amount of light you were supposed to get. And this individual didn't have any signs of damage to their skin. So it's not the level between safe and unsafe, and people can be in spaces with far UVC acting continuously as long as they're installed correctly.
In fact, you make me recall, I think it was probably one of the first one-take podcasts I did. I do this midweek one where I review a paper, and there was a paper out of Leeds actually looking at far UVC. That was the one, was it? Yeah. So, that was really interesting because they looked at the impact of 1 bulb versus 2 bulbs and certain air change rates and so on. And it was really interesting to see the efficacy at different levels. Very good. And practically at an operational level, because this is something that we experience a lot in the ventilation world, is the degradation or the installed performance of devices. Is there a practical test that you can walk into a space and measure the effectiveness of the lamps that are there? Or is it really a laboratory only test? Can a technician with the right piece of equipment go into a space and assess whether it's been effectively installed, that it's got the coverage that it needs, and it's working to the right wavelengths, for example?
Yeah, so there are devices out there. They're a little expensive right now. So most of the companies are the ones having to go in and do the spectroradiometer scans. But you can go with an electronic device and measure the exact output of these lamps in these spaces. Additionally, if you want to do it yourself, they have those dye indicator cards. I've got a bunch of them in my office drawer here, which will have a color change, just like if you're doing a chlorine test for your pool. And as long as it was in a specific range, you know that it's acting as it could. But the difficulty with that is the amount of time you put it in the lamp affects the dose, and the dose affects the color. So that one's a little more just to make sure it's on kind of test. Whereas the other, you can get more.
Got what you paid for. Have I just got a purple light, or have I actually got far UVC test?
Yeah. But that is one of the benefits of the fact that the light doesn't happen outside, is these dyes aren't reacting to really anything else. So
yeah, really interesting. But you can, the manufacturers are going around with a piece of equipment that can actually say, right, okay, we're getting the right level of light in these areas and treating the room the way we expect it.
Exactly. So every one of our lamps are turned on and then checked before we send them out.
Yeah. It's been really fascinating. Should give you a chance to plug as well a little bit Lit Thinking, because it's a really innovative company and one right at the cutting edge. Explain. So this was a company that was a startup based in this university that you were working in. Is that right?
Actually, the startup started in 2022. This one was out of Orlando, Florida. So we actually work really closely with the University of Central Florida, and have done some research projects with both of them. And we're now building our own semiconductor facility up at Cornell. So check us out there in a couple of months once we get everything up and operating. But the Visium device that we have created has gone through all of the rigmarole. It was it was the first to be certified for continuous use by UL for this 8802 standard. We are working to make sure that we are up for all of the different regulations as they start rolling out. So we're EPA registered. All of our devices have their own efficacy data. So not based on things that were published. We trapped them in a chamber. We put things on glass just to make sure that we checked all of our boxes, because we really want to be leaders in this space. We want to be examples of good actors who are trying to do the right thing for a new technology, because, you know, one bad actor will give us the whole field a black eye. And we're working really hard to make sure that we can be that trusted partner to talk about shepherding this whole new field forward. So a lot of what I do with these Visium devices is took it through UL, do these ongoing case studies where I get to go on location and show that in every shape of room, you can have a benefit. So recently I went to the pig farm, and I went to the COVID clinic, and pretty soon I'm going to go back and do a locker room for an ice skating rink. So it's just to show people and get them involved and excited, because it can be any space. It can be your spaces that could be a little bit cleaner. So it's something that we work really hard to get people involved. And actually right now we're doing a Vanguard program. So we want it to go into places where there's a good need, but maybe there's not funding for it. So we'll set up your first room and do a test to show you exactly what good it can do. And that way we've been able to reach a couple more corners where maybe it would have taken a grant to get there.
And where's your market? Is it just North America at the moment? Are you looking at exporting internationally? What's the market for your product?
Right now, we're primarily US based. There have been a couple of small projects that have been in other countries, but because it's a new technology and the rules and regulations aren't defined everywhere, and sometimes they change their mind, we're trying to do it on a kind of a case-by-case basis for everywhere out of the US.
Do you see any risk in the market at the moment for people as well? Like what you're seeing out there on the horizon? Like you say, you know, I'm reluctant to call it emerging technology. I don't know how many centuries we're going to be talking about UVC before somebody says it's an established technology. But do you see, not bad players necessarily, but are there risks being taken out there with products and technology when it comes to far UVC? Because as you said, you know, it only takes a couple of bad actors to create a bad smell in the sector. And I don't hear about that. It's nothing that I've seen. But do you see elements of that on your horizon? Does it worry you?
I'm not too worried, because a lot of the bad actors kind of make themselves known by not having good supporting data or installing them in a way that you kind of look at and go, huh. So one of the reasons we picked ceiling mounting is because it is the safest way to deploy into a space, because most people aren't staring directly at their lights, and eyes are the most sensitive parts of our body. So normally our light's hitting our eyebrows, it's shaded for our eyes, so there's less exposure risk. So we're thinking about how to limit risk every way we can, whereas some device companies are shining it straight forward, and that makes me nervous, even though I know it's very, very low risk because they've done the studies on eyes. That's the last thing I want to do, is give people a reason to question. Because some of the reason that these standards were set so low to begin with is there was something installed incorrectly in a hospital, and a couple of nurses got some fairly severe sunburn for the original traditional UVC devices. Now, while sunburn isn't a risk that we anticipate because of the studies that we have already done, that changed the trajectory of UVC for 50 years, and that's the last thing we want to do.
Yeah, baby steps and in the right direction. And it's all about optics with startups as much as it is anything else, or new innovative technologies. Yeah, no, for sure. Well, it's a fascinating area and an absolutely fascinating topic to talk about. It's been Brilliant. I've learnt loads, as I'm sure the listeners have. We'll of course put links in the show notes to both Lit Thinking and any other studies that you think might be. In fact, I put that lead study in the show notes as well again, because I think it's a really interesting third party look at the efficacy of the technology. I remember speaking to the authors about a year ago, and they were really excited about the results of it, considering they had no skin in the game really, and they came away from it going, "Oh my god, this stuff is amazing," So hence it was one of the first ones I put up on the one take. So we'll put that in the show notes as well. Janet, thanks a million for your time. It's been brilliant chatting to you.
Oh, it's been a pleasure. Thank you for having me.
Thanks for listening. Before you go, can I ask a favour? Before you shoot off to whatever it is you have to do next, or even the next podcast, if you enjoyed what we've been talking about here, please do spread the word and let's keep building this community. And don't forget to check out our sponsors in the show notes: Ultra Protect, Eurovent, Erico, ACO, Farmwood and Zender, all amazing companies, and without them this podcast would just simply not be possible. Check them out in the show notes at airqualitymatters.net, and do check out our YouTube channel. All this content is there, plus much more. See you again next week.